All right, let's jump right in. Have you ever stopped to think that maybe the entire universe, from the biggest galaxies right down to the tiniest molecules inside of us, is governed by one simple, elegant idea? Well, today we're going to explore that idea, symmetry. And what's even cooler, how life itself figured out that the secret to thriving is knowing exactly when to break the rules.
Okay, so take a look at these two. At first glance, they seem like they're from completely different worlds. On the left, you've got a molecule like sulfur hexafluoride. It's a perfect model of balance and order. And then on the right, you have a protein, which just looks like a complex, chaotic tangle of stuff. I mean, they couldn't be more different, right?
Well, as it turns out, they are deeply connected. What links them is this kind of hidden language of symmetry, a set of rules that tells us how molecules can be spun around, reflected in a mirror, and inverted, all to check if they end up looking exactly the same. It's like the secret architecture of the entire molecular world. So if we really want to understand the story of life, we have to get a handle on this molecular language first. Let's break down the fundamental, shall we?
At its heart, it's a pretty simple idea. We say a molecule has symmetry if you can do something to it, like spin it on its axis or hold it up to a mirror, and when you're done, it looks exactly like it did when you started. It's completely unchanged, or, as a chemist would say, it's indistinguishable.
So let's check out the basic moves, the operations we can use to test for this symmetry. As you can see here, we can rotate a molecule around an axis, we can reflect it through a kind of mirror plane, or we can invert it through a single point right in the center. These are really the fundamental building blocks of symmetry.
But some of these operations are a little more complex. They actually combine two moves into one. A great example is the improper rotation. So first, you rotate the molecule by a certain angle, and then, step 2, you reflect it through a plane that's sitting perfectly perpendicular to the axis you just spun it on. It's a two-step combo.
And this is where it gets really interesting. This quote illustrates a key point. Just think about a methane molecule. If you only rotate it 90 degrees, it doesn't look the same, and it doesn't have the necessary reflection plane by itself either. But if you do the 90-degree rotation, and then you do the reflection, bang! Suddenly it looks exactly like the original. The symmetry only shows up when you combine the steps.
So we've got these really elegant, fundamental rules of symmetry, which brings us to this fascinating paradox when we look at life itself. Why are the molecules that literally run our bodies, like proteins, so beautifully, asymmetric? It turns out this isn't some accident. It's a feature, not a bug.
It all kicks off with what's called the primary structure. The easiest way to think about it is like a string of beads. Each one of those beads is an amino acid, and the specific order of the beads, glycine, then alanine, then valine, that sequence is what defines a protein at its most basic level.
But here's the thing. That chain does not stay straight for long. Based on all the different interactions between the amino acids, it starts to fold in on itself, creating this incredibly intricate and very specific three-dimensional shape. And that shape, that's what gives the protein its job. And this folding process actually creates multiple layers of complexity. First, you get these little local structures like spirals called alpha helices and folded-up planes called beta sheets. Then the whole chain folds into its final, what we call tertiary structure. And sometimes you even get multiple folded chains that come together to form a quaternary structure, just like we see in hemoglobin.
Okay, so here is the crucial point. The amino acid building blocks themselves actually have a handedness. They can exist as either left-handed or right-handed versions, perfect mirror images of each other. And life, for reasons we are still trying to figure out, almost exclusively uses the L, or the left-handed version. And that one choice guarantees that the final folded protein will always be asymmetric. So, what happens when this fundamental rule gets broken? Well, for some bacteria, learning to use the other hand is not just a party trick. It's a key to survival and a really powerful weapon in this constant molecular arms race.
So while basically all of life is building its proteins with these L amino acids, some clever bacteria have figured out how to use their mirror image, the D amino acids, in their own structures. And they use this molecular trick as a pretty powerful tool for defense.
And this clever little trick has some seriously powerful applications for bacteria. By swapping in D-amino acids, they can make their cell walls totally unrecognizable to antibiotics, like vancomycin, which makes the drug useless. The bacteria that causes cholera actually uses them to make its own cell walls stronger so it can survive starvation. And what's really fascinating, we can turn this trick back against them by using cocktails of D-amino acids to break up the protective colonies or biofilms that they form.
This molecular arms race is absolutely fascinating, right? But the whole thing depends on being able to actually see these changes happening, and that raises a huge question. How on earth can we possibly watch something so tiny and so subtle happening at an atomic scale? How do we see these molecules in action?
Let's get even more specific. How can we possibly see something as fundamental and as fast as a single electron just jumping from one atom to another? I mean, this is absolutely key to understanding how these molecules actually behave. Well, we use some highly specialized tools. And one of the most powerful is something called Mossbauer spectroscopy. You can think of it like a super-powered microscope that uses gamma rays to get unbelievably precise information about the world right around a specific atom, like an iron nucleus.
This technique is so sensitive that it can tell us if electrons are localized, which means they're kind of stuck on their home atoms, or if they're delocalized and moving so fast between atoms that we can't even tell them apart anymore. When they're localized, we see separate signals for different iron states, but when they're delocalized, those signals just blur together into one single average signal.
And what's really wild is how the molecule's environment totally changes this behavior. Scientists took an iron complex and dissolved it in benzene. In that solvent, the benzene molecules literally stacked up against the complex, holding it totally rigid and static. And as a result, the spectrum clearly shows two separate signals. The electrons are basically frozen in place, localized on their own iron atoms. But then, they took the exact same molecule and put it in a different solvent, one that didn't lock it down. And suddenly, it became way more flexible and dynamic. And just look at that spectrum. At room temperature, those two signals merge into one averaged peak. And that is stone-cold proof that the electrons are now delocalized, moving freely between the iron atoms. This discovery is just such a powerful reminder that in the molecular world, shape is absolutely everything. The tiniest, most subtle change in a molecule's structure, or even just its environment, can have these profound consequences. By understanding the rules of symmetry, and maybe more importantly, when and how life breaks them, we can unlock secrets that affect everything from medicine to materials science. It really makes you wonder, if a simple flip from left-handed to right-handed gives bacteria a superweapon, what other incredible secrets are just hiding in plain sight, waiting to be revealed by the shapes of the molecules that build our entire world?